The overall goal of this procedure is to measure the complete carbonyl content of various biomass-derived materials by titrometric methods.
This method gives insight into the reactive carbonyl compounds contained within biomass-derived products such as pyrolysis oils and the effect that those compounds have on the aging of bio-oils. This method uses smaller sample sizes, less toxic materials, is more precise and more accurate than previously established methods.
To begin this procedure, prepare the reagent solutions as outlined in the text protocol. Dry a sodium carbonate primary standard in an oven at 105 degrees Celsius overnight. After this, allow the sodium carbonate to cool to room temperature. Weigh 100 to 150 milligrams of the dried sodium carbonate into a metal pan. Transfer it to a titration vessel and record the actual weight. Then add a stir bar and enough water to cover the pH electrode bulb and the junction. Using an automatic titrator, titrate with the hydrochloric acid solution to the end point, which is the inflection point on the titration curve. Repeat this titration twice using a new sodium carbonate sample each time to obtain a total of 3 endpoint values. To begin preparing blank A, add 0.5 milliliters of DMSO to a 5 milliliter vial with a spin vane. Next, add 2 milliliters of the prepared hydroxylamine hydrochloride solution and 2 milliliters of the prepared triethanolamine solution.